All right, good afternoon, everyone, and welcome to today's NAVBO webinar sponsored by the Education Committee. I'm Dr. Amber Stratman from Washington University in St. Louis, and I'm moderating today's session. So I'm pleased to be welcoming Dr. Julia Mack from UCLA, who will present her talk, Mechanisms of Endothelial Cell Resilience. Before we begin, I'd like to remind you that all NAVBO activities are easily found in the calendar of events on our website. And please be sure to register for some of these upcoming events. Throughout the presentation, please type your questions in the question box, which is to the side panel, and the questions will be answered at the end of the talk. So today's webinar is being recorded and archived on the NAVBO website for future use. So I want to tell you a little bit about Dr. Mack before we begin. Dr. Julia Mack is an assistant adjunct professor in the Division of Cardiology at UCLA. She received her BA in chemistry from the College of Holy Cross and her PhD in inorganic chemistry from UCLA. Working in industry for the materials division at Teledyne Scientific, she designed functional composite materials for aerospace and biomedical applications. However, the emerging field of regenerative medicine sparked her decision to return to UCLA in 2011 as a postdoc in the Department of Molecular Cell and Developmental Biology. During her postdoc, she studied the role of Notch1 in arterial homeostasis and collaborated with engineers to design 3D bioreactor systems to control fluid flow. In July 2018, she started her independent research group at UCLA in the David Geffen School of Medicine, where she is an active member of the cardiovascular research team. She was awarded a Career Development Award from the American Heart Association and serves as a mentor for the AHA STEM Goes Red program and the AHA Hispanic Serving Institutions program. Her research team studies mechanobiology of endothelial cell signaling using high-resolution imaging techniques and multiple in vivo models to interpret the role of blood flow forces in cardiovascular health. So we're really excited to have you here today and to hear about your upcoming and ongoing work. So welcome, Dr. Mack, and I'm going to let you take over.
Thank you, Amber. Thank you for the introduction and for hosting this webinar this morning. I'm excited to share a story about endothelial cell resilience. And I'm excited to generate hopefully discussion and comments and questions at the end of the talk, of course. So I'll start by talking about kind of what this resilience idea, it really was kind of timing here when I started my laboratory in July of 2018. It was around the same time that NIH had initiated this trans NIH resilience working group. And this really struck a chord with me, this idea that in response to some challenge or stressor, how a body or a tissue or an organ or a cell would resist or respond to recover to maintain homeostasis was an area of research that was kind of just generating this idea of, how do you, boost resilience to improve vascular health or health in general. And I urge you to take a look at this commentary that Yulin Gao and Zarina Gallus wrote just a couple years ago in ATBB, where they're really pushing for this idea of studying endothelial cell resilience as a way to discover potential diagnostic and therapeutic strategies to improve all-body cardiovascular health. So, coming to the field, as Amber mentioned, I came with a very different background in chemistry. And, starting as a vascularology as a postdoc, just really, getting exposure to the field, I was recognizing that very often the field focused on disease risk factors and pathogenic mechanisms. And while that's all well and good and very important, I wondered, what if we shift our focus instead to studying what's happening in the absence of disease? And in that way, would that be a way that we could define or identify cell resilience factors, which would be the ability of the cell to cope with environmental changes to, like I said, maintain homeostasis or to recover homeostasis? So this isn't really necessary for this audience, but in any case, in terms of cell resilience, I'm focused in cardiovascular health, and specifically I study the endothelial cell, which obviously is the most important cell. But there are, as you know, the innermost lining of all your blood vessels. And when I talk to students and I tell them why I'm studying endothelial cells, it's because by definition, you can't have a blood vessel without an endothelium, right? That innermost lining, that is what a blood vessel is. Obviously, smooth cells and parasites and other cells are important, but that endothelial lining is essential to forming and maintaining what a blood vessel does. All right, so in terms of what I study in terms of mechanotransduction, I really focus on blood flow, right, which is this frictional force due to the flow of blood across that endothelial surface. And it's been well known in the field that the type of flow or type of shear stress can influence the endothelial cell phenotype. And just to kind of show something that's been out in the literature for many years now, I recognize this is 1999. If we look at, what's known in terms of this normal shear stress, which actually is quite a large range, but atheroprotective flow, which is high arterial shear stress, you get low proliferation and low apoptosis, you have low expression of adhesion molecules. These cells tend to be anti-inflammatory, hence atheroprotective flow. Whereas in regions of very low flow, for example, low arterial shear stress, you get the exact opposite. You get more proliferation and apoptosis and expression of growth factors, and low expression of vasodilatory molecules like nitric oxide, as well as antioxidant enzymes. So going to shear stress, right, we talked about flow, wall shear stress over the endothelium. This is a very simplified cartoon and inaccurate, really, because there's no one flow sensor. The field has looked for one, but really I think it's a whole dynamic process in terms of how the endothelial cell or the endothelium as a tissue, how it responds to shear stress and flow. And what I wanted to point out with this very simplified, naive cartoon here is that the cytoskeleton really plays a key role in transmission of forces throughout the cell. And we'll kind of get to that as I advance the slides. But that signaling really is transmitted via a lot of kind of changes in the cell. So I like to start with this video because, well, I just love it. I do a lot of live cell imaging, and it's going to show the response of the endothelium to the application of the onset of laminar shear stress. So this is a monolayer of human aortic endothelial cells that we cultured overnight so that you can see that they're a confluent monolayer. And then what we're going to do is I'm going to start the video, and it's going to be the onset of 20 dynes per centimeter squared. So that's going to be, flow is going to be moving from right to left across your screen. So I'll start the video. And I hope you can see that there's a lot of dynamic changes that happens in response to this onset of flow. You see a lot of proliferative events. You also see, actually that was like a pruning event, actually that cell didn't make it. But you can see there's a lot of proliferation. There's actually a lot of migration. And cells tend to move actually upstream. You can see them kind of heading off the side of the video at this point. But you see a lot of activity, right, of the endothelium in response to the application of flow. And now what I like to do is I'm going to fast forward closer to the 24-hour mark. And what I hope you can appreciate is that this monolayer of cells looks quite different, right? There's much less proliferation. We don't see a lot of proliferation at this time point. The cells look a lot quieter. And I think this kind of leads to this, terminology we have of quiescence, right? Less active. And very simply put, right, you can see morphologically, these cells look very different at the end after 24 hours of flow than they do at the beginning. So this slide is just here to really emphasize that here's your static cells cultured. They kind of resemble this cobblestone morphology that people will often refer to, because I remember when I first started in the lab, they're like, oh, well, endothelial cells should look cobblestone-like. And they do in static culture. However, if you apply flow, and this is laminar shear stress, 20 dynes per centimeter square, which is mimicking the human aorta and the descending aorta, you really get a dramatic morphological change, a dramatic morphological elongation and alignment with flow. So going back to our vessel of choice, we really like to study the aorta because in this same vessel, in the same monolayer cells, there's variable shear stress. This is due to variable blood flow patterns as blood is pumped out of the heart and then goes through the arch and then down the descending aorta. What you have is you have regions, like I talked previous slides before, of kind of more laminar shear stress or normal shear stress range, compared to regions of very low shear stress or very high shear stress in the arch. And what this sets up is regions where the endothelial cells tend to be atheroprotected, as we talked about that, anti-inflammatory, and regions where the cells tend to be more prone to inflammation and atherosclerosis. And that's in the regions of very low shear stress or very high shear stress. So going back to where I started with resilience, I wondered, well, can we study the cells in this descending aorta where they're typically protected? Maybe by studying how they signal or what they do, right? How their phenotype, what enables their phenotype. Maybe we can identify and define resilience factors. So in my laboratory, I do use multiple systems. I tried to avoid mice, but you can't. It's very hard in vascular biology. But we do use mice, and they are a great tool. What we can do here very simply is just take the aorta out of a wild-type mouse, and we can open it en face and stain for B-cadherin and ERG. ERG is a nuclear marker, so we know we're looking at the endothelium. And if you look at the endothelial cells in the lower arch, which is this region of low shear stress, and compare them to the endothelial cell monolayer in the descending aorta, where it's this high laminar shear stress, you can see very clearly that morphological difference, right? You can tell very clearly what type of flow these cells were experiencing. In the lower arch, they're more cuboidal, that cobblestone morphology actually, right? Versus the descending aorta where they are elongated and they align in the direction of flow. I also love that, right? I can look at the endothelial cells and I know which direction flow is going. Excuse me. So what we've done here is we can apply segmentation masks and then quantify, for example, the aspect ratio. And what you see is that you get over about a threefold increase in the cell aspect ratio for endothelial cells in the high flow region. In my laboratory, as I mentioned, I like to do a lot of live cell imaging. And so the Abidi pump system is a very useful tool that we utilize in order to visualize endothelial cell signaling and behavior under flow. And in this case, we like to use the Y-shaped slide, which allows us on the same monolayer of cells to look at a region of low shear stress and a region of high shear stress. And if we compare these two, the low flow is probably around 4 dynes per centimeter squared, compared to high flow is around 20 dynes per centimeter squared. And comparing the morphology of these cells, we can recapitulate pretty nicely the cells in that lower arch region where they're more cuboidal, cobblestone shape, versus the high flow of the descending aorta where the cells are elongated, and again achieving that threefold increase in aspect ratio. So these are two of our systems we use. Now, going to actually work, I bring this because this is really the other component of why this project and what we're studying is during my postdoctoral training in Luisa Rila Aerospace Laboratory while she was still here at UCLA. One of the things I saw, and I just thought it was so striking, is that the full-length Notch1 receptor would polarize or accumulate at the downstream end of flow-aligned endothelial cells. And we actually did some experiments that are in this paper from 2017, where we reversed the flow and the full-length Notch1 receptor would reaccumulate on, again, the other end downstream of flow. And so this set up this idea, actually my American Heart Association for Development of War was based off of this idea that, simply put, maybe in the presence of flow and cell body elongation, maybe the two ends of the endothelial cell are different. Simply put, just like that. So we've continued that work in my laboratory to really try to kind of characterize the endothelial cell in response to the cell body elongation. And what we find is that there is a cell asymmetry. The way we have done that is by measuring different biophysical properties. In this case, we've done AFM. This is tapping mode, where we're just getting the height profile of a single flow-aligned endothelial cell. And I like to point this out. It's so amazing. You have this giant nucleus, right, around 5 microns in diameter. And then the ends of the endothelial cell get really quite thin. We get down to, I don't know, around 200 or 250 nanometers in thickness. But in any case, we're interested in these two ends, the upstream versus the downstream end. One readout of biophysical property we've done is using force mapping using atomic force microscopy. And you can see that we can get a force map across the downstream versus upstream. And we actually find that one end, the downstream end, tends to be stiffer than the other end. Another readout is membrane fluidity using Lardon dye. And so this actually also indicates that one end is different than the other end. Again, this downstream end of the cell tends to be more rigid compared to the other end that tends to be more fluid. Another readout of differences across the cell is actually lipid composition. So this is staining for Bodipi C5 ganglioside, GM1, which is an indicator of raft-associated or raft-like lipids. I try to avoid raft, but in any case, we do see an asymmetric distribution where one end tends to have a higher accumulation of these raft-associated lipids. Another readout we see is actually by F-actin staining, very simply. It just struck us that, by looking at the monolayer, there be regions at the downstream end where there be more accumulation or higher density of F-actin staining. I was interested in the cell surface, right, because of, I guess, Notch1 was one thing, but another thing that got me interested is, it's known that endothelial cells have a high, express high degree of caveolin-1, right, and Cav1 as well as Cavin-1 are both really important scaffolding proteins to form caveoli, which are these invaginations, right, in the endothelial cell plasma membrane. And so I was curious whether or not there might be an asymmetric distribution of caveolae or caveolin-1, for example. So we did staining. In A panel, we're using the mouse descending aorta. And here on the right side, I just show that what we do is we segment the cells, only the full-length cells, right? Because we only want to quantify the distribution where we can see the upstream and the downstream end, right? And then we can, we basically divide the full-length cells into equal length thirds. So we define as upstream, mid-body, and downstream. And then we essentially just quantify the mean intensity of fluorescence for, in this case, caveolin-1. And what we find in the flow-aligned endothelial cells of the descending aorta, there's a higher accumulation or expression of Cav1 at the downstream end. And things tend to be a little bit more striking in vitro, and you can see that in panel B here, where by staining for caveolin-1, you see the high density or accumulation of Cav1 staining at the downstream end and not the upstream end of the cell. So that's quantified here, where we're quantifying the mean intensity from these 3 different regions of individual cells. So really, this is kind of pointing to this idea that maybe in response to cell body elongation, the presence of laminar flow, things accumulate on different ends of the cell, or at least there seems to be an accumulation here of caveolin-1 at the downstream end. So we've gone on to kind of in more careful imaging to kind of characterize what these membrane domains look like. And so what you can see here is a higher resolution imaging of cells. These are flow-aligned endothelial cells, and this is what I'm talking about, this F-actin. It almost looks like F-actin basket or bundling. I don't know, it's a higher, it's like a meshwork there, which is really interesting. And right there is where we see this high density of caveolin-1 staining. So, as we started looking at this more and more over time, it really started to appear to look like a membrane domain because we're seeing this only at one end of the cell. So one way of kind of quantifying this, because you have to quantify everything, right, is we define like a cluster index. And what we find is that we get more caveolin-1 clustering at the downstream end than we do across the other, regions of the cell. So I also was talking about signaling, right, in terms of resilience factor. And one thing that is well known to be increased in response to laminar flow is nitric oxide production. That's well known. You go from static to flow or disturbed versus laminar flow, there's an increase in nitric oxide production as well as an increase in phosphoinos, which is the active form and the enzyme required for nitric oxide production. So we stained for phosphoinos. This is for the serine 1177. And you can see very clearly that this is caviola 1, where I'm just kind of doing this rainbow coloring. You see high density of cav 1 tends to be at these, what we're calling tips, or these domains. And this is really what struck me, is that we found also kind of correlated with that high density of caviola 1 staining, these focal accumulation of phosphoinos. So in theory, this is the active enzyme. So this is where there is nitric oxide potentially being produced. And we find that it's interesting, there might be focal production of vasodilatory factors at these domains. And this gave me some excitement here because, I wanted to make a statement about, okay, maybe these are signaling domains and something is happening that's important for vascular function. And nitric oxide production is obviously a very important one. So are these signaling domains? We think so. Another readout of signaling actually is calcium activity. And because of the role of calcium and calmodulin in eNOS inactivation and activation, we actually decided to look at calcium activity across the monolayer of endothelial cells. This is the very first video, one of the very first videos of once we had transfected our human aortic endothelial cells with GCamp, so that they're transfected with a calcium, a fluorescent calcium reporter, so we could do long-term imaging experiments. This is what we saw. So here's these cells. So just so you know, all the experiments we do are 48 hours of flow. So they've been kind of flow conditioned. And then we apply flow continuously. What you can see is this is recording a snapshot, of their activity in the presence of laminar flow. And so I hope you can see all the sparkles. It really struck me when we first saw this, and I'll play the video again, that there were these beautiful sparkles across the monolayer. A couple things really intrigued me. One being that it wasn't all cells. There seemed to be things going off like, Christmas lights across at different time points. And also striking, it wasn't whole cell body oscillations, It seemed to be focal sites, localized sites in endothelial cells where calcium was entering the cell. So I'll move to the next slide to zoom in to really convince you of this. So here's a zoom in of one cell where I've highlighted in green the whole cell, so you can see the full length. And then in black, I'm defining as this downstream tip. And then red is the remainder of the cell body. And so I'll play this video, and I hope it convinces you that we really are getting focal or localized calcium entry at this downstream end of the cell. And so this plot up above is just representation of the mean intensity of the GCamp, the green signal over time. And this is for a cell that was recorded for 15 minutes. And it's interesting that we're getting this persistent calcium oscillations, calcium entering, and then it closes, and then so we're getting these focal bursts of calcium entry. So what we've done is taken many monolayers. We do lots of imaging. We've done a lot of segmentation. And again, we only segment the full length cells because we're really interested in the upstream versus the downstream, But across the monolayer cells, when we've imaged for 30 minutes of calcium activity, we find approximately half of the cells are active, meaning during that time frame of 30 minutes, about half of them exhibit some kind of calcium oscillation or calcium entry. Of those cells that are active, over 70% of them exhibit localized calcium entry at this downstream end of the cell. So this focal entry that's localized only at the downstream tip. This video I like to show because I find it pretty convincing. So again, I'm going back to that Y-shaped slide from Abidi, which I think is a useful tool to look at a gradient of shear stress. So at that back end, we're going from, again, this is all laminar flow, but we're going from low shear stress, about 4 dynes, to 20 dynes. And this gradient really picks up very quickly. And you can tell even if you look that here is right where the cells are experiencing that low flow, and they're not elongated, or there is no, you can tell there's no flow directionality, right? They're not elongated with flow. However, if you go down further, you start to see the cell body elongation and alignment. So I'll play this video, and what I hope you can see is that very little activity is happening in that low flow region. All the activity really is where the cells are experiencing this high laminar shear stress. So play that just so you can see that gradient. It really does seem to be right dependent on the shear stress level. So I'll move forward to just kind of show, this is that same experiment, but we're just showing that the calcium activity in green is highlighted, in yellow, sorry, is highlighted, and the low flow, very little activity, and high flow is where all these speckles are. These are the summation of the calcium activity over the 30 minutes of imaging. And if we do a zoom in of low flow versus high flow, cell shape really seems to be at play here. And we've actually found that there is a positive correlation between the aspect ratio of the cell and whether they, and the tip activity. So there does seem to be this dependency, potentially, or correlation with cell shape, elongation, higher aspect ratio, and more tip activity. This I like to show side by side. This is, again, the same monolayer cells on that Y-shaped slide where we're able to image the low flow and high flow simultaneously by going back and forth. And yes, they're both playing. I'll do, let's see, I'll back up and play it again. But it's really quite striking. Not only do you see a very dramatic difference in cell shape, right, the elongation of the cell in high flow, but there's so much more activity than low flow. So, what does that mean, right? To really get at, because somebody asked, is this a mechanosensitive domain? Can we say that? And I think we can, because of a very simple experiment, which I don't know why it took us a while to do this, but it's so simple. Sometimes the simplest things you forget to do. But in any case, this is an experiment we did flow, right? And we're recording 30 minutes of flow. You can see these calcium oscillations at the downstream tip. So this is that downstream tip. We stop flow, and those calcium oscillations go away. They're dormant. If we turn back on flow, the oscillations come back. So we really do think that flow is required for signaling at these domains. Along the way of presenting the work, you get great questions from the audience. And somebody asked me whether or not cell elongation would be sufficient for signaling in these domains. And so we actually initiated collaboration with IBIDI of a tool that I think they now have commercialized, but you can essentially generate patterned slides. And so this is an RGD pattern of lines, thin lines, and the goal with this was just to get the endothelial cells to elongate along these lines in the absence of flow. So we were able to achieve the same aspect ratio, but in the absence of flow. And so here's flow aligned cells. You can see a monolayer. This is the pattern static cells where they're elongated as well. But what was interesting, I find, is that when we stained for caveolin-1 phosphoenos, caveolin-1 was still asymmetrically distributed. I was actually kind of surprised by this, meaning that potentially it's the active cell body elongation, not necessarily flow, that that causes this asymmetric distribution of caveolin-1. However, in the absence of flow, we did not see these focal accumulations of phosphoenos. So down here in panel C, you can see we're showing that, in fact, it does appear that caveolin-1, in response to cell body elongation, the absence of flow can accumulate on either end, either left or right, right? There's no flow directionality here. But also, I think interesting is it's not significantly different between left versus right, because I think flow would give that kind of directionality. We don't have any directionality here. In terms of why we're not seeing those focal regions of phosphoenos, I think it's because we're not getting this calcium activity that we see in the presence of flow. So this is we're just recording the GCAMP cells, and we don't see this activity in the absence of flow. And so panel E, we're showing that by staining for phosphoenos in elongated no flow versus low flow versus high flow, you see this increase in phosphoenos. And this is well known that high flow would increase phosphoenos, But what hasn't been shown is that potentially, what we think is happening is that it's being activated, or its activity is occurring at focal domains, or these membrane domains in endothelial cells under flow. All right, so then, what is this activity? The first thing we did was actually a simple experiment to convince ourselves that I thought it was probably some channel, right? Some calcium channel. That it was calcium coming from outside in. And so to do this, we just added EGTA to the flowing media, and we saw that, in fact, we could suppress these calcium oscillations, indicating that we do think it's a channel that calcium's coming from outside in. And then around, of course, you do Google searching, right? And I found this paper out of Mark Nelson's lab, first author Swapnil Sankosara, who I just met last week at NAVBO meeting. Very excited to meet him, because this work from 2012 in Science, in vivo, showing that mouse mesenteric arteries had these elementary calcium signals, is what they called them. And in the paper, they refer to them as TRPV4 sparklets. And when I read this and I saw this, I was like, I think that's what we're seeing in our monolayers of cells. I think these are TRPV4 sparklets. And so we did the simple experiment of adding GSK205 to our flowing media, which this is a specific TRPV4 channel antagonist. And what we saw is that when we do that, we block that calcium activity. So we really do think it's TRPV4 dependent. I'll also say that we did other antagonists to confirm it wasn't piezo. It really is TRPV4. So now, is TRPV4 asymmetrically distributed? Maybe the channels, I thought, again, some kind of naive thinking, maybe TRPV4 is just at these downstream domains, and it's not. Makes it more interesting, right? If we compare low flow versus high flow, there's no difference in TRPV4 expression. So it doesn't seem to be flow regulated in terms of expression of the channel. And it's not polarized. It's not accumulated at downstream end. It's kind of ubiquitously expressed across the whole cell surface. And it's not really flow responsive in the traditional way that a lot of times we think about things, right? KLF2 and KLF4 are well-known flow responsive genes. TRPV4 does not change in response to static versus flow. Here, static versus flow, you can see eNOS, also that's well-known. TRPV4 kind of just generally the same amount. So it's not really, the expression of the TRPV4 channel doesn't seem to be changed by flow, but its activity is. So what is regulating that? So we think that it is this association with caveolin-1 at the downstream end. And to get at this, we did a proximity ligation assay. So we were able to stain for and visualize the physical interaction of TRPV4 with caveolin-1 using PLA. And so this really, I think, beautifully shows that in response to high flow, there is more TRPV4 caveolin-1 association by these puncti, these purple puncti that you see here. And in response to the high flow region where the cells are more elongated, we have a higher association of TRPV4 and caveolin-1 at that downstream end. So we really think it's what's around TRPV4 channel that's regulating its activity. So, in biology, I feel like you have to kind of mess with things to really prove something. So we wanted to get rid of these domains. And the way we did this was we used kind of a brute force method, but it worked, and beta-cyclodextrin, which is known to chelate cholesterol. And in that way, it's known to collapse caveoli structures. And so what we did was we did flow alignment, and then we added M-beta-cyclodextrin to collapse the caveoli, and we kept the cells in LPDS, lipoprotein deficient serum. So what we did here is we just messed with the domains, right? You can see on the control cells, you have these focal accumulations of CAV1 at downstream end, and in cholesterol depleted, we don't, we lose that. So we mess with the domains. In response, we see less nitric oxide production, and we don't see that TIP activity. However, we know the TRPV4 channels are still there, because if we add GSK101, which is a known agonist of TRPV4, we see these the TIP activity come back. So TRPV4 is still there, but it's what is surrounding or around TRPV4 channels that's modulating its open probability or its channel activity. All right, again, in biology, you have to mess with things to convince everybody that it's an important thing. So what we did was we inhibited TRPV4 signaling or activity in the presence of laminar flow, and we did this for 2 hours, and then we did RNA sequencing. And this was really nice that it showed us that the cells look very different from each other by PCA analysis, and a whole bunch of different genes were affected. But what we started to see by analysis, and this is qPCR data, a validation of several genes that we saw changed by the RNA sequencing data, is that in the absence of TRPV4 signaling, we started to see an inflammatory signature. So you see E-selectin, CCL2, VCAM, ICAM, IL-6, NF-kBIA, RELA, all of these were increased, meaning that even though the cells are in the presence of laminar flow that's atheroprotective, right, and anti-inflammatory, if we inhibit TRPV4 signaling, TRPV4 activity, they become inflammatory. And so this was some of the pathway analysis that we looked at, really indicating that there was an inflammatory response. And one of the primary upstream regulators that we identified was REL-A. So we think that this is somehow moving through NF-kappa B. So we did some further experiments just to convince ourselves. Again, always flow alignment for 48 hours. And then in this case, we added DMSO or GSK205 to inhibit TRPV4, and then we looked at the cells. I like to visualize them, right? And so in the response, in the absence of TRPV4 signaling with GSK205, we get less nitric oxide production as evidenced by DAF-FM staining. We see an increase in reactive oxygen species, which I think is interesting. This is as quantified by cell ROX expression. And then this is for this and panel D is 4 hours. We start to see the emergence of adhesion molecules like ICAM1 and E-selectin. And this is in panel E, actually only 2 hours of inhibition of TRPV4. Very clearly, we see NF-kappa B, P65 nuclear localization, right? So in the absence of NF-kappa B pathway, the NF-kappa B P65 is cytosolic, but in activation of the pathway, you see nuclear localization, and we see that really quite clearly. It's also interesting, I find, that it's only certain cells. Anyway, all right, so then we wanted to confirm that this is important in vivo. So we used wild-type mice, and we injected them with GSK205 or DMSO. So we wanted, granted this is systemic, but in the analysis we did, we looked at the descending or aorta en face, so that we're actually looking at the endothelial cells, the arterial endothelial cells. And in the absence of TRPV4 activity with GSK205, just like in vitro, we see an increase in inflammatory gene expression like VCAM1 expression, as well as nuclear P65. So finally, going back to where I started with resilience, can we say that these signaling domains that are mediated by TRPV4 activity, can we say that this is a resilience mechanism of the endothelium? And so the way to get at that is we asked, can we boost this signaling activity to improve the response after exposure to an inflammatory agonist? And so, yeah, can we enhance endothelial cell resilience? So the experiment we did flow alignment for 48 hours. Then we took the cells off to static, and we did experiments, and I can confirm that in static state we don't have that calcium activity. Remember, we did flow, static flow. So in static state, the calcium activity is absent. So what we did in static state is we hit the cells with TNF-alpha. So this is known to be an inflammatory agonist. But we either added DMSO or GSK101. And the addition of GSK101, we confirmed that we would get TIP activity. And so in G is staining for these cells, and DMSO, you can see very nicely they're anti-inflammatory, right? No nuclear P65. If we hit them with TNF-alpha in the presence of DMSO, they go nuclear, right? There's all the nuclear P65 staining. Now, if we add GSK101 to activate these TRPV4 signaling domains, we see an attenuation of the inflammatory response. It's not 100%. Also, I find interesting that seems to be certain cells. In any case, we see an attenuation in the inflammatory response. And we confirmed that by staining, as well as by qPCR showing that certain things aren't changed, like CD31 and ITGB1, but we see a reduction in NF-kBIA. This is the inhibitor of NF-kappa B. And we see a reduction of VCAM, CCL2, E-selectin, and TNF. So, of course, everyone asks about piezo, and I wanted to share this. We did look at activation of piezo in response to the inflammatory agonist. And the activation of piezo channels with YODA1 does not attenuate the inflammatory response. In fact, if anything, it augmented that inflammatory response. And I think this is powerful data and information, because I think what this points to is the idea that it's how calcium comes in and where that really is important for downstream consequences, right? Because calcium, right, is the ubiquitous, like, can do everything, right, and nothing, I guess, can do everything. But how calcium comes in and where it comes in, I think is really important in the endothelial cells. So finally, just to also hone this in on DMSO versus TNF-alpha plus DMSO, where we get all the nuclear P65. TNF-alpha by activating TRPV4 with GSK101, we see attenuation of that inflammatory response. That's here in the dark red. However, if we activate piezo with YODA1, we do not see any attenuation or suppression of inflammation. So finally, actually, we did this same experiment using an in vivo model of LPS. So these are wild type mice that were treated with LPS, a fairly low dosage of 1.5 meg per kilogram. And we either added DMSO or GSK101, similar to our in vitro experiment here, to activate the TRPV4 channels. And then we harvested the aortas after 4 hours. And we did some on-phos staining, which is in panel H, and we did mRNA. This is from a luminal flush, so we're really collecting arterial endothelial cell enriched RNA. And what we find is that I thought this was really cool, that NOS3 was increased with the activation of the TRPV4 channels, which is in line with previous reports that TRPV4 is really important for vasodilation. And we see the suppression of inflammatory genes, REL, REL-A, REL-B, CCL2, and IL-1 beta. So again, this idea that activation of TRPV4 at these signaling domains, even in vivo, we do see an attenuation of the inflammatory response. So it seems to be a way that the endothelial cells can boost their resilience, right? And boost their response to this, in this case, inflammatory stimulus. So here is just the heat map of gene expression. And then finally, on-phos staining, we always like to do to confirm what we see by either protein or by RNA expression. Here we see that in the descending aorta, the vessels that were treated with GSK101 to activate TRPV4 had less VCAM1 expression and less nuclear P65. So this actually was really nice when, an in vivo experiment actually recapitulates what you do in a dish. And, of course, I think part of this is that we always do everything with flow. So finally, I wanted to make sure I leave time for comments and critiques and questions. I think that what we are showing here is that there is a mechanosensitive signaling domain in arterial endothelial cells that promotes resilience. In this way, right, this polarization or accumulation of specific lipids, specific membrane-associated proteins like caveolin-1 sets up these domains that enable localized calcium entry, in this case through TRPV4 channels, at these domains to activate eNOS and suppress, I think, nitric oxide and suppress reactive oxygen species. And in this way to promote vasodilation and also prevent inflammatory signaling. So I think TRPV4 and these signaling domains are one conduit through which anti-inflammatory signaling is achieved in arterial endothelial cells in the presence of high laminar flow. Because if we inhibit that TRPV4 activity, we start to see an inflammatory endothelial cell, even in the presence of high laminar flow. And it's one where we have reactive oxygen species and less nitric oxide production and NF-kappa B signaling. So we believe that potentially this might be a therapeutic tool in the future to target vascular inflammation, and maybe a way that we can identify to boost endothelial cell resilience and to improve cardiovascular health. And so in summary, we believe that endothelial cell body elongation induces a lateral asymmetry of the cell surface in this way to establish polarized plasma membrane microdomains. These domains are polarized at the downstream end of arterial endothelial cells, and they are mechanosensitive. Right? Remember, if we stop flow, we don't see the signaling activity. And we believe that these microdomains facilitate TRPV4 mediated calcium entry focally, right, locally, in the presence of flow to activate eNOS and to inhibit inflammation. And so finally, I would like to acknowledge funding sources from American Heart Association, always so important. We all need funding. Also the UCLA Cardiovascular Discovery Fund that is supporting my postdoc, Seung-Gu Kong, and UCLA Diabetes Research Center, as well as a list of collaborators here, as well as abroad. And I'd like to give a very important shout out to Seung-Gu Kong, who really took the project when he started with me just over a year ago and really took it to the next level. Seriously, it's very impressive work, and really we've been a fantastic team, and I want to say a big thanks to him and to the other members of the Mack Lab and to all of you out there for listening this morning, or this afternoon, or this evening, wherever you are.
Excellent, excellent.
So right on time, right?
Right on time, and perfect too, because we have a ton of questions.
Good, that's
so
why I wanted to leave it.
Yeah, I'm going to hop straight into them. So the first question came up fairly early in your talk, and I think you addressed a lot of it, but I'm going to extrapolate from it a little bit. The question was, by shear stress, do you look at laminar or oscillatory flow? And I think you hit on that you mostly look at laminar flow. But do you think you see differences in endothelial cells in different vascular beds? And if you think, if they have different developmental origins, do you think you would see differences?
Yeah, great questions. Okay, but yes, I will confirm pretty much everything we're doing is laminar flow. I will tell you we are also looking at disturbed oscillatory flow now, and I can tell you that if we change from laminar flow to disturbed flow, the signaling changes. So the type of flow is really important, but yes, pretty much everything I reported here in this presentation today is all laminar flow. And in terms of differences of signaling across different vascular beds, a question that I've been asked multiple times and something we are very interested in looking at. I can say from Swapnil's work, they looked in mesenteric arteries. They also have looked, I think there's TRPV4 signaling, Mark Nelson's lab has looked at in the brain, the cerebral vessels. We have really not looked diligently across many vascular beds in terms of origin, right? We have not done that. What we are doing right now is taking different endothelial cells, which these are cell lines though, right? But we're looking at microvascular versus vein versus arterial because we are interested. Is this a signaling feature of only arterial endothelial cells? I don't know yet, but we are hoping to address that.
Yeah, very cool, very cool. Okay, our next question is, what happens to inflammation if you activate TRPV4 in low or no flow conditions?
If we activate TRPV4 with GSK101 in no, so are they, I guess, for the question, I'm wondering, are they already flow aligned or are they just, static? So if they're just static cell and we activate TRPV4 with GSK101, those domains aren't present. So we don't, but we did, to be honest, we didn't look at any kind of inflammatory we didn't investigate whether there was a difference in inflammation and static cells that were activated for TRPV4.
Okay, do you think you could force domain presence if you activate TRPV4 artificially?
I don't know, because I think that it's not just TRPV4, but it's what's surrounding it, right? That's required for it. I think that's what's regulating its open channel probability there. So caveolin and different lipids. We have some data now. We're looking down lipid pathways, which I get scared about because I don't know enough about lipids. But I do think that specific lipids are probably modulating its activity. And so in some ways, I think the cell body elongation enables the presence of these domains, the stretching of the cell. And I think it's probably a big component is related to the cytoskeleton.
Mhm.
And I was just talking with someone a couple of weeks ago of, how important, and I didn't think about this until they brought this up, of, the cell surface, that membrane is not just the plasma membrane, but it's the underlying cortical, right?
Mhm.
So that it's kind of like the cell skin. And it's not just membrane, but it's also the underlying cortical actin there that is so important. So I think it's all connected. And so that's why I think we see the accumulation of actin, the kind of more fibrous F actin there. But yeah.
Awesome. That kind of takes us into our next question.
Yeah.
They say, this is a fantastic investigation. Well done. Do you believe this TRPV4 pathway is dependent on continuous laminar flow over elongated cells? What would you expect to see if the flow direction was disturbed, reversed, or otherwise altered after your laminarization?
So I can tell you, I kind of mentioned this. We have done those experiments, and it's super cool that in the presence, so we have laminar flow going, and then Sunguk has changed it where now it goes disturbed back and forth oscillatory, and the cells not only change shape, right? This is known, right? That flow is required to maintain that cell body elongation, but the calcium signaling changes dramatically. What we see in response to this oscillatory flow is like this big increase in calcium throughout the whole cell body. And then somehow the cell figures out that it's not good to have too much calcium, and it suppresses it. I don't know how. I'd love to talk more with Swapnil and other ion channel people, because I don't really know all the mechanisms there. But somehow endothelial cells, the cells are smart. They figure out how to turn off things that are dilatory to them.
Yeah, they're way smarter than us, right?
Yeah, yeah.
Okay, the next,
I try to learn from them.
yeah. I'm just going to keep going through because
Okay.
we've got a bunch of
Okay.
questions. So, okay, so the next one says, thank you very much for your fantastic presentation and exciting work. Do you have any insights into whether there is a correlation between these laminar flow calcium channel activation and changes in von Willebrand factor coagulation responses of endothelial cells?
Ooh, I have not looked at VWF. Have not looked there. We could, yeah. Um, Stay tuned. I can take a look and let you know, but we haven't looked at it yet.
Wonderful. Okay, terrific talk. Many thanks. I understood from the literature that CAV1 inhibits nitric oxide synthase. How do you reconcile that with your observations where CAV1 clustering seems to increase NO signaling?
Excellent question. Because yes, in the literature, I will say that it seems to be a little murky sometimes. But yes, in the literature, it's been thought that CAV1 sequesters eNOS to inhibit its activation. But we are not seeing that. We are seeing at these domains where there's CAV1, we are seeing phospho-eNOS, and this is the active form of the serine 1177. It could be both, right? The CAV1 does inhibit, but it's the calcium entry there that I think is what's activating it. So that is my hand waving way of saying this is what we see. But I totally agree. In terms of the literature, there's literature that's reported that CAV1 is inhibitory of eNOS activation. But all I can say is what we're seeing currently is we're seeing there is a focal activation or focal accumulation of active eNOS there.
Yeah,
At the
maybe
downstream end.
maybe this will be one of those things that falls out, between arteries and veins or different
Right.
origins or something like that too.
Right. Yeah. I mean, I think we do need to look more into it. I mean, at this point, we kind of just report what we see, but I do think understanding that we haven't gotten to really to the mechanism, right, of understanding how is eNOS activated selectively there. We're correlating with TRPV4 and calcium entry and CAV1.
Yeah, there's so many things, right?
Yeah.
All right, next question. Brilliant work. I was wondering if the resilience of endothelial cells and brain microvasculature is somehow connected to endothelial mural cell communication. If in the case of hemorrhage, will this resilience still work? And do you know if there's any endothelial cell, mural cell crosstalk that's affected?
Yeah, these are great questions. And I would recommend they look at some of Mark Nelson's work, because he does a lot of work in the cerebral vascular realm in terms of actually TRPV4 signaling. So I would suggest looking into some of his work. But I can tell you, I actually am interested. We have zebrafish now, which is, Amber, I still need to talk to you about some fish work. But
Sure.
with zebrafish, what I want to do is compare calcium signaling activity, for example, in the dorsal aorta versus the arches versus the brain. And I think the brain is a really cool, because it's this beautiful network, right? And so my idea there is for us to look at this calcium activity, and I have a collaborator that we're interested in modeling it. In our case, we have GCAMP only in endothelial cells. So we're only going to see calcium activity across the vascular network from calcium signaling in endothelial cells, but absolutely, I think it's so important, the crosstalk between the two muscle cells or parasites, those mural cells and endothelial cells. We aren't currently looking at that, but I'm sure there's other people that would be interested potentially in collaborating or
Yeah.
going down that road.
Yeah. Okay. Next question. This is kind of a technical question. So, what motivated the 48-hour long assay versus a shorter time point?
Good question. Um, we find, but yes, 24 hours, we do see nice elongation, right? I showed that video at the beginning. But I was really hoping to get to quiescence. And we found that at 48 hours, we really saw a suppression of like proliferative pathways and proliferation of the cells. And so that's why we stuck with 48 hours. And pretty much all of our experiments are 48 hours. It also worked well of alignment over the weekend. And then we do all our experiments. Anyway,
I like pragmatism in our
I'm just
experiments.
telling you, yeah, I'm an open book. But yeah, no, 48 hours we do see, we believe, really nice quiescence.
Yeah. Okay. We probably have time for 2 more questions, and
Okay.
then any questions we don't get to, we are going to send to you, and you can directly
Okay.
answer to people.
Okay.
Okay. So the next one is I may have missed this, but is the downstream end of the cell more rigid in terms of membrane? And if so, would the TRPV4 channels be more difficult in terms of opening, as the membrane rigidity may prevent that? And then the next question, well, I'll let you address that first, and
Yeah.
then have a follow-up question.
Yeah. So we do find, I think the fluidity, right, is more rigid. By lard and dye, we see specific lipids seem to accumulate there. We also see certain lipids not there. I can tell you we've been looking at PIP2 as one thing, which might be something more related to signaling. But yeah, it does seem that that end is more rigid by the kind of biophysical properties we measured. But what that means in terms of TRPV4 and open channel probability, I don't quite know. But I think it's about what's surrounding it that may be regulating it, right? That I think Swapnil has shown some really elegant work of CAV1, but there's something else that also is regulating the TRPV4 opening. So it could be less about the rigidity of the membrane, if we think about it that way, but more about what's surrounding it that enables its opening, for it to open more, I guess, efficiently or easily.
And then the follow-up question to that was the MBCD chemical is fairly nonspecific.
Yep.
Have you considered other lipids that are known to modulate TRPV4?
Yeah, so PIP2, where we've been doing, actually after talking to Mark, we've done some experiments with PIP2 that look interesting. Yes, there's other lipids involved, and there's a project that we're collaborating with the people here at UCLA on modulating the lipids a little bit more elegantly. Absolutely. And beta cyclodextrin is a brute force hammer. And yeah, we just suck out cholesterol. The cells don't like that. But it was our way to get rid of the polarization of CAV1. That's all we wanted to do. But now, in collaboration with Peter Tonchina's lab and JP Kennelly here at UCLA, we're gearing up for more elegant experiments of modulating lipids in the membrane and looking at this calcium signaling activity.
Wonderful. Okay, last question here is fantastic work. Do you think laminar shear stress can induce vessel elongation by inducing endothelial cell proliferation, or do you find any evidence to support that?
That's laminar shear stress for promoting proliferation, for growth of the vessel.
Yep.
Yeah, we don't typically look at development, right? But I mean, in response to laminar flow, there is a major increase going, but this is the thing I will have to say that sometimes I sometimes get confused too on this is I think the onset of shear stress, how the endothelial cells going from static to flow, I think is very different than cells that are conditioned, right? And are
Mhm,
maintained under flow.
mhm,
So I find I've been trying to get at whether there could be a cell memory of whether they had experienced flow and what type of flow. I
mhm,
don't have any answers to that. I think that's an interesting, like more philosophical question. But in response to shear stress, yes, there's an increase in migration, proliferation, um, but whether or not, and during development, right, flow does turn on, and then you get a whole bunch of vascular development happening. So maybe flow going from the flow turning on induces maybe a lot of that, and then over time, maybe the cells then settle down and then they take over and they become quiescent and the
mhm.
vasculature is formed and everything's happy, healthy, and you keep it like that.
Yeah, some more work to be done, right?
Yeah, yeah.
All right, so it's right at I guess
Yeah,
2 o'clock Eastern now. So I'll
look at that.
say thank you again, Dr. Mack, and then we will yeah, have questions sent to you and you can respond to people directly. So I hope everybody enjoyed today's webinar. Look for more information on future events. Our next webinar will be on December 7th with Dr. Joyce Bischoff from Boston Children's. And thank you for joining us today.
Thank you.